Excuse me, everybody. We are going to start, I'm going to call it our lunchtime cabaret act. David, is that appropriate? I'd like to introduce you all to David Weinstock. David's the VP for Discovery Oncology at the Merck Research Labs, where he leads all of the Discovery Oncology teams. Prior to joining Merck, he was the family chair of preventive medicine at Dana-Farber. He's also worked as an associate physician in medical oncology in Brigham, as well as a professor of medicine and pediatrics at Harvard Medical School. So David's going to give us his perspective as we munch along on our lunch. Thank you.
Well, thank you very much. I was at the last CancerX meeting and I made some provocative statements. And so on that basis, Andrew asked me if I would follow up with a keynote lecture. He also guaranteed that I would be compensated twice what the other speakers are compensated. I'm going to talk about some of the evolution of my thinking as I went from being an academic for 14 years to industry. And specifically, this question around evolution or revolution and where we are. And so, first of all, I'm an employee of Merck. I have financial relationships with some companies that I started or helped start, and also with Merck. I've received research support from my previous laboratory from many companies. And all the opinions that I'm voicing are my own. They do not represent any opinions of Merck, MSD, any affiliates otherwise. So this is venture capital investment in the United States, as far as I can tell, number of deals over the past several years, as well as the value of those deals, as you can see, came down a bit in 2022. And I think those of you who are in biotech and VC know this quite well. And it totals in the 40-something billion dollars per year. This is how hard it is to make a drug. So you start with thousands of different compounds, and then you move forward hundreds into preclinical studies, and then you move forward several into clinical trials to get to one FDA-approved drug. And as far as I understand, the reason why we're here, at least to some extent, is the idea that if you just made this bigger, you would get to more approved drugs. If you just put more money in, then you would fund more research, and you would get to more drugs. And I think that's a brilliant hypothesis around the idea of hedging by taking a collective approach. It is predicated somewhat on the hypothesis that when you make it bigger, you'll maintain a probability of success for each individual project that's similar to the probability of success that we have now. And not that we're already in a meritocracy where we're skimming the cream and everything below it is milk, but that there's plenty more cream. And it's just not, it's not ultimately being capitalized. I'm mixing many different metaphors, but into drugs. So how hard is it to make a drug? I think most of you know it's very hard. I'm going to talk about 6 of the challenges that we face as a field for oncology discovery. And that's in the midst of tremendous successes over the past decade. So the first is, how do we predict the future? And I love this quote from Wayne Gretzky, that I skate to where the puck's going to be, not where it has been. So as I just showed you, when you start with an idea, if I start today with a new idea, or I see a paper in Nature from a brilliant investigator like Mike Yaffe or Keith Flaherty or something, and I say, well, we should make a drug that capitalizes on that, 7 to 10 years to market. So what's going to happen in all that time? A lot. So one of the first things we do is go through competitive intelligence, and this is an outdated version of competitive intelligence for what's going on in PSMA-targeted therapies. And this is just what's announced, and this is just clinical. So there are hundreds of different agents that are being developed by many different pharma against each of these targets. And so you may think you have a really good idea, but it turns out in the context of where the field's going to be, where the puck's going to be by the time your drug is ready to be approved, it actually may not be a good idea. The other is that you don't know what you don't know. And a lot of the biotech bubbles up from an incredibly intellectually vibrant academic world. I just give this as an example. This paper just came out in Nature Genetics. It's an example of tremendously collaborative work done by centers across the country, very similar to the type of things that Keith talked about. And you can see that the Stand Up to Cancer Mark Foundation cohort brought together 393 patients with non-small cell lung cancer who were treated with some kind of PD-1 or PD-L1 therapy, and then performed extensive genomics. And we're talking about how important RNA-seq is in this context. Well, I can tell you that Merck has a database of 50,000 patients with exomes and RNA-seq treated with Keytruda and followed for outcomes. But we don't share it with anybody. And that's a real challenge. It's a challenge because it would be financially unwise for us to take that out of the competitive space and make it non-competitive. We spend enormous amounts of money to collect that. And it's a challenge because there are precedents where companies have shared that type of data. Academics have done analyses, and there's one very famous one where an academic group published a safety signal that they derived from the data that was in a very underpowered study, and the FDA added a black box warning to that drug based on that analysis. And subsequent analyses have not borne that out, but the company felt that there was an enormous amount of market share that was lost because of that. So there's also a reflexive desire to protect your own franchises that I think limits some of that. Nonetheless, I do want to talk about opportunities, I think, for trying to mine those kinds of data. And if we have that kind of data, you can imagine Roche does too, and BMS does too, and so on. The other thing is, it's just hard to predict the future. So this is weather.com. All right, and this is the high temperature forecast for weather.com for Washington, D.C., beginning the actual day of the prediction and then going 1 day ahead, 2 days ahead, 3 days ahead, and so on. And each colored zone is 15% of predictions over a period of time. And there are two really striking things. One is, once you get to day 8, there's about a 30-degree span in the ability to predict what the high temperature is going to be. 30 degrees for the high temperature in the city at day 8. So obviously prediction is very poor. And even more than that, what I think is striking is it doesn't get any worse after day 8. So basically at day 8, you've maxed out sort of the chaos theory effect on your ability to predict. And it doesn't get any worse after that. So we're trying to make predictions not only about what the market's going to be like in 7 to 10 years, but now you add on the Inflation Reduction Act, and it's another 9 or 13 years until negotiations, and we're talking decades of predictions ahead. It's just incredibly challenging. All right, so I want to tell you some of my best worst ideas, or worst best ideas, you can decide, where we as an academic laboratory decided to develop approaches and face multiple challenges, and I learned through those challenges. So on the left, you see an example of Burkitt lymphoma. On the right, you see Dennis Burkitt, who first described this back in the late 1950s. This is a disease that affects both people in developed countries and in primarily Africa, where it's almost always associated with Epstein-Barr virus. It tends to cause these massive lymphomas that are in the jaw. They used to be 100% fatal. They're incredibly rapidly growing, possibly the most rapidly growing tumors on Earth. These cells can double every 8 hours. And under the microscope, pathologists describe this as a starry sky appearance. So what you see is just a sheet of tumor cells. Each one of these little circles is a tumor cell. And then each one of these big cells is a macrophage. So this cell is known to be infiltrated with a large number of macrophages, these immune cells that are known to just eat up cells. But nonetheless, the lymphoma continues to grow and grow and grow. And the idea of the starry sky is that the macrophages are supposed to be the stars in the sky. So Burkitt described in the late 1950s and then into the early 1960s remarkable responses to endemic Burkitt lymphoma in children treated with cyclophosphamide. So a very simple molecule that now costs less than 20 cents a day. And he even described kids who were cured with a single dose of oral cyclophosphamide. That's almost unprecedented across all of cancer. And we have not really understood why that is. One of the major challenges for trying to understand this is that cyclophosphamide is actually not an active drug. It's metabolized in the liver into an active drug and then further metabolized in multiple different ways. So it's quite complex. So all the studies of cyclophosphamide really need to be done in vivo systems. And so my laboratory has been one of the groups that has developed a very large number of patient-derived xenografts, including some xenografts that we in-licensed from Novartis. It was about 800 total xenografts across solid tumors and hematologic malignancies. And many of these maintain certain tropisms, like they still grow. You can see in the lymph nodes in that spinning mouse. You can see in its sternum and its hip, that's bone marrow growth. You can see in the skin, in the brain, and so on. we turned this into an open source repository called the Public Repository of Xenografts. And we provide these at cost to academic centers and to many, many industry partners around the world. I left all this behind at Dana-Farber. This still exists, but you can see we've provided models to over 600 institutions around the world now. So this is work that we did with Mike Heeman, who is a Koch investigator, really brilliant guy, and a very good friend. And some work from his lab, initially looking at cyclophosphamide in models of Burkitt lymphoma and other lymphomas that are similar, had shown that cyclophosphamide can recapitulate that really remarkable effect. So this is basically a femur from a mouse that's been xenografted with a Burkitt-like lymphoma. And now you take a slice of that. And everything you see that's sort of in the purple-blue is lymphoma. So it is just, the bone marrow cavity is just completely filled with lymphoma. If you give the mouse the maximum tolerated dose of doxorubicin, another chemotherapy, no effect, just completely filled. If you give the mouse the maximum tolerated dose of cyclophosphamide, then this brown is actually staining macrophages. So within 48 hours, tumors 99% gone and completely replaced by macrophages. These are, if we stain them, they're not proliferating, so we know that they've actually come from outside in the mouse and they've come into the bone marrow cavity. And we also know that if you add an antibody that binds to these cells, it works even better, because then the macrophages see that antibody-coated tumor cell that's dying and they just chew them up. It's just candy. So we designed a clinical trial to test this, and the antibody that we used was one called alemtuzumab, which was marketed by Sanofi as Campath. And we had the trial all written, and we went to Sanofi and we said, would you provide the Campath? And they said no, because they had pulled Campath from the market because they found that this drug that was not making very much money, if you dosed it about 30-fold lower, it worked very well for multiple sclerosis. But they didn't want to charge 1/30th of the charge, right? They kept the same price per gram. So if they want to increase the per gram price, they can't then have it available for researching cancer where you need 30 times as much, because the price would now be $6 million a year. So they just pulled it from the market and said, we're not going to do any more research on Campath. And that's financially a very reasonable decision. As an academic researcher, I had apoplexy and sent all kinds of emails that I should not have sent, and called the CSO at the time, who I've subsequently mended relationships with, and threatened to go to the press, and other things that I would never do now. Please don't do those to me. And we did actually get the drug. And so we moved forward with the study. This is the first patient who we treated. This was at the Dana-Farber. So this is a patient with widely disseminated lymphoma. You can see everything that's black on that PET scan over there except the bladder is tumor, massive tumor, kilos of tumor. Patient, over the course of time, had received multiple cycles of salvage chemotherapy that had basically no effect, had one cycle of what we called Cycam, had this really dramatic effect, went into an almost complete remission, but then decided at that point didn't want further therapy. There's no doubt this was a toxic approach. Nonetheless, had a really remarkable response. Second patient actually had a complete response. So this is a CAT scan. You can see the liver up in the top left, 3 days before therapy. All that sort of homogeneous color around it is actually fluid that's filled with tumor. There's a bunch of lymph nodes. 35 days after this one cycle of chemotherapy was in a complete response. This was the white blood count, 300,000. Upper limit of normal is usually around 10. So this was all tumor, completely gone. Patient got an allogeneic stem cell transplant. It's now about 5 years out, presumably cured of their therapy. Patients number 3 and 4, I obviously won't show those because they didn't do as well. And then we were about to do patient number 5, and CAR T cells became the standard of care. So the puck moved. And the therapy that we were proposing to give was so immunosuppressive that basically would have prevented patients from being able to get CAR T cells. And so a very reasonable decision by our lymphoma group was they just weren't interested in pursuing this anymore. So it goes. All right, so that's challenge number 1. Challenge number 2, how do you buck the trends? How do you get over FOMO? So these are 2 studies of pembrolizumab or Keytruda, and I obviously choose those studies, but there are remarkable ones of nivolumab and atezolizumab and other PD-1 blockers as well. But these are two that really, I think, move the needle. So the first is in microsatellite unstable colorectal cancer. These are patients with metastatic colorectal cancer. Prior to this, the standard of care was chemotherapy. And you could see 90% of those patients within 2.5 years had progressed, and almost all of those patients were dead. And the ones who hadn't progressed, they weren't cured. They probably just had very, very slow-growing tumors, and they managed to maintain some kind of haven't progressed yet. You give single-agent Keytruda, and 40% of those patients are cured. Cured. It's unbelievable. It is true revolution. Why does this happen? We have no idea. We have no idea why Pembro or other PD-1s do this. CTLA-4 doesn't, TIM-3 doesn't, LAG-3 doesn't, IDO-TIDO doesn't, STING doesn't, adenosine 2A and 2B doesn't, CD73 doesn't, and so on and so forth. We don't know, but it does. And if you combine it with chemotherapy, something that was actually really poo-pooed by the field, because the thought was the chemotherapy is going to kill the T cells, and then the T cells won't work, and so on, you get even better outcomes. So this is Keynote 189, first-line non-small cell lung cancer, also associated with a very significant cure rate. Definitely not this high. It's gotten worse over the course of 5 years, but nonetheless, much, much better than chemotherapy. And Keytruda is now approved in over 30 different cancers. It's a $25 billion a year drug. It has absolutely, along with other PD-1s, changed the treatment of cancer, rewritten textbooks. So I hear a lot of, I think we should make another immune checkpoint inhibitor, right? And these are some of the immune checkpoints that have already been targeted, and then Gordon Freeman or someone else discovers another immune checkpoint inhibitor, another company spins out, and people say, make another one of these because it might be the next Keytruda. Does anybody know what this is? It's a lemming. So, like lemmings to the sea, we have followed this path for the last decade. And it has cost hundreds of billions of dollars. And it just has not worked out. And they were perfectly reasonable hypotheses. But at this point, I think we've got to have an enormous amount of skepticism about just turning on a T cell more in a nonspecific way. Nonetheless, there are many, many companies. And if you look at this really nice review of the clinical challenges of immune checkpoint inhibitors that was in Cancer Cell, you can see two-thirds of the active trials in cancer in 2020 were T cell modulators. Yet, if you look at the immuno-oncology drugs with novel mechanisms of action, so this is non-PD-1, non-CTLA-4, that were approved, 0 in 2019, 0 in 2020, 0 in 2021, and 1, the LAG-3 from BMS, relatlimab, in melanoma, and it was a pretty small benefit. So, that's depressing. I also wanted to talk about synergy, because this is something that comes up over and over and over again. Our drug is synergistic. We've shown our drug is synergistic in vitro. We've shown our drug is synergistic in vivo. I'm very much on a soapbox here, but the beautiful work done by Adam Palmer and Peter Sorger at Harvard Medical School, and also done by Emmett Schmidt and Kong Chen internally at Merck, has shown that synergy is a preclinical phenomenon. We just should not use that term anymore. And it doesn't mean you can't ever come across something that's synergistic. But they have shown with every possible combination that you can think of that's been tested, where it's 2 drugs and you've got the PFS curve, and each of the 1 drugs has been tested with a PFS curve, that if you just do a very simple mathematical calculation for what you would expect, giving 2 drugs instead of 1, no synergy, that the PFS curve for the 2 drugs looks exactly like what you would predict. So the immuno-oncologists say, yeah, sure, that's true for signaling molecules and chemotherapy. It can't be true for immuno-oncology because we make cold tumors hot, and then the PD-1 comes in, and that changes everything, and so on. Okay, so this is the nail in the coffin as far as I'm concerned. This is an analysis of all the studies with immune checkpoint inhibitors, where you had a PFS curve for the immune checkpoint inhibitor. You also had a PFS curve for the single agent it was being combined with. That could be a chemotherapy, a target inhibitor, or a PD-1, or something else. And you can then make an expected progression-free survival, right? And this is for all time points that have been studied, and this is just for the studies that only looked at 12 months. That's why there's 2. But let's focus here for now, for all time points that have been studied. And then you could say, okay, what's the observed progression-free survival for that study? And as you can see, everything is on the line. If anything was synergistic, it would be over here. There's nothing. And I want to note, each one of these dots is not a patient. It's a trial. It's a combination trial. This is tens of thousands of patients in combinations. Despite the fact that many phase 2s, as you know, are somewhat biased, right? Because they select certain patient populations, might do a little better, and so on. Still, nothing is above. Many things are below, possibly because of interactions, but also possibly just because 2 drugs is more toxic than 1, so more patients are likely to come off the trial then, and they do worse in the combination than you would predict. This doesn't mean combinations are a bad idea. Combinations are a great idea. These patients still benefited from the combinations, but they didn't achieve synergy. So I think we look at all in vitro and in vivo synergy with enormous skepticism. Okay, challenge number 3, expecting to fail and then learning from it. This is a wonderful quote from Daria Hazuda. This is at our center in Cambridge. In the back, you can see it's a missed opportunity if we fail to learn from what doesn't work. Daria was the head of infectious diseases at Merck for a very long time, helped develop Crixivan and other really pivotal therapies. One of the names that you've never heard but really made an enormous difference in the world. So this is KRAS. I am incredibly excited about KRAS, but these are the described mechanisms for clinical resistance to KRAS inhibition. All right? So there's a wonderful JCI paper that came out of a rapid autopsy of a patient who was treated with a KRAS G12C inhibitor, and they biopsied multiple different sites of disease that grew, and they found 5 different mechanisms of resistance in the same patient. So when we think about overcoming resistance and appropriate combinations and so on, this is what we're up against in many situations. And so we're very skeptical of strategies that are going to overcome resistance to a particular therapy because we just know that almost always the processes are extremely complex. The other is around robustness. So we see a lot of this kind of data. This is actually internally generated Merck data, so I'm not picking on anyone here, where we developed some kind of new drug that we want to combine with PD-1 blockade. So we take a tumor, we put it into mice, usually on the flank, and then we treat with an isotype antibody that's basically a control, or we treat with the anti-PD-1, you can see in red, they do better, or we treat with the new drug, the 4464, they do better than the isotype, and if you treat with a combination, you see this beautiful combined effect. Right? And then you can see the individual mice here, and in the combination, 11 out of 15 mice had complete responses. So we should move this to clinic. This is the kind of data that's in most data packages. This is 1 tumor. 1. We don't ever treat patients or establish a new FDA standard of care based on 1 patient. So why would we do it based on 1 mouse tumor? And these are technical replicates. That's all they are. So in contrast, this is beautiful data from Daiichi Sankyo. So Daiichi Sankyo developed what became the drug of the year last year in HER2. It was called DS8201 at that time. It's an antibody conjugated to a cytotoxic payload that binds to HER2. And instead of doing 1 tumor, they did 34 tumors across multiple mice of different histologies, of different levels of HER2 expression, of different resistance to the payload. And then they compared it to TDM1, which is the Gilead Kadcyla drug that was the standard of care at the time. And then they could show very clearly their drug just works better, right? And then they presented this data, and then they developed the drug, and then they got it approved, and then they presented additional data at ASCO this year. And this is an actual shot from ASCO of the standing ovation that occurred. Larry was there, I'm sure. How frequently do you see jubilant oncologists? Yeah. So it was Larry's colleague who led the study. And still, this isn't curing patients, but it's a remarkable advance, and it's just much more robust data that's being presented. So I love this quote from George Box: all models are wrong, but some are useful. He's a mathematician, and I think since all models are wrong, the scientists must be alert to what is importantly wrong. And it's inappropriate to be concerned about mice when there are tigers abroad. So we need better models to make better drugs, and I think there have been some beautiful presentations already in this meeting about trying to develop those models. Models are tools for asking questions. So when someone says, my model represents cancer, you have to clarify in what way. What question does this allow me to ask in a way that I think is going to be more predictive? And I love this paper. So liver metastases have been known to be a very significant problem for a long time. So this is in melanoma, this is in non-small cell lung cancer, it's also true in other cancers, that patients with liver metastases do worse than patients who have metastases to other sites, whether it's lung or lymph nodes or even brain. So why is that? Well, we don't know still, but these people did a really brilliant experiment where they took this cell line, MC38, it's a mouse cell line, so this is in an immunocompetent mouse. It's known to be very responsive to PD-1 blockade. And they xenografted it onto the flank of these mice. And this is the standard thing, just the flank, right? So here you're not studying liver cancer, or you're not studying colon cancer metastatic to the liver or lung to the liver. You're studying flank cancer. We don't treat a lot of flank cancer in human beings, but that's what we use to study. So if you treat with the anti-PD-1 or L1, you get this beautiful response where the tumor gets smaller. All right. Now if you do the experiment again, and you xenograft onto the flank, but you also orthotopically xenograft into the liver. So now you have synchronous disease in the liver and on the flank. And then you treat with your anti-PD-L1. You see it doesn't respond. But this isn't the liver. This is the flank, right? I would have guessed the flank would respond, but the liver wouldn't respond. And so if you take out these flank tumors before you treat them and you actually look at them, they have a completely different immune infiltrate in the flank tumors, just because there happens to be a liver tumor at the same time. And it looks like what the liver is doing is very much soaking up and completely changing the immune response to the tumor throughout the body. So what this tells me is that we can do as many organoids and other things as we like. If we want to ask a specific question about liver metastasis, we probably need a model that recapitulates that. All right, and then one thing that really changed in my mind when I went from being an academic to being in industry. As an academic, I focused on efficacy. Does it make the tumor get smaller? Could I possibly cure people? As in industry, I realized how important it is to actually focus on therapeutic index. Therapeutic index is the most important thing in all of drug development, in my opinion. And I'll explain why. So therapeutic index is the difference between what you can get in terms of efficacy and what you get from toxicity. And you want it as broad as possible. And if you follow that to a ridiculous endpoint, basically you can say this. The most toxic substances on Earth, things like actinium-225, right? One molecule of actinium-225 can kill probably 4 or 5 cells. One atom, right? Or polonium, or bleach, or something like that. Those kill cancer cells spectacularly well, right, in a dish. But if you give them to human beings, they kill the human being too, if you just give it, right? Because it has no therapeutic index. So that's the most toxic substance on Earth. The least toxic substance on Earth, water, right? If you put cancer cells in pure water, they will all die. But you can't give water as a therapeutic, because if you gave enough water to kill the cancer cells, you'd kill the human being also. So basically, any range of toxicity from the molecule itself is not what's important. What's important is the difference between its effect on the cancers and its effect on the rest of the human. And I think for the scientists or some of the finance people, you probably don't think about these things, but you might hear about them, grade toxicity standards. So what you hear is, oh, it was only 15% grade 3 diarrhea, or something like that. So it's tolerable, well-managed side effects. Okay, well, let's go through. Grade 5 is death. Grade 4 is almost death, right, ICU. Grade 3 is probably hospitalization required. So if you have grade 2 toxicity, it means you are sick up to the point where you have to go in the hospital. So you can imagine, if you've got 12 months of grade 2 toxicity of any kind, that's really not tolerable. And grade 1 is, you might be able to work, you might not. So when we think about these toxicities, and especially as we think about benefiting patients for as long as possible, we actually need drugs that have grade 1 toxicities, maybe some grade 2, but these grade 3, 15 to 30% are just not going to fly. One example of a drug that's like that is belzutifan, or Welrig, which is one of our drugs. This is based on work, Nobel Prize winning work from Bill Kaelin, and then developed by Peloton, not the bicycle company, the pharmaceutical company, and we acquired them in 2019. This is a brilliant development of a molecule that can actually block a protein-protein interaction that prevents a transcription factor, HIF2 alpha, from doing what it's supposed to do. It really only affects cells that have a genetic defect in something called von Hippel-Lindau. So in children who are born with von Hippel-Lindau disease, or in clear cell renal cell carcinomas, where 90% of them have von Hippel-Lindau mutations, the drug is unbelievably effective, and it causes almost no toxicity. There's definitely some anemia on target. There are some other grade 1 toxicities, but this is a drug that you can take for years. This is the waterfall plot in the renal tumors. So each one of these is an individual tumor. You obviously like it when everybody is below the line. And so I hope this becomes the cornerstone of future renal cell cancer therapy, that you can then add PD-1 and VEGF receptor inhibitors and other things too. So those are the kind of drugs we get really excited by. What Merck can do in the context of a drug like that, so I talked about a relatively small population of patients, but boom, right? 4 phase 3 trials within 2 years. A number of phase 2 and phase 1 trials in every kind of combination with our drugs or other people's drugs to just explode the market really in renal cell cancers. And so that's a great opportunity. Obviously, small biotechs can't do this, and that's one of the reasons why they tend to partner with big pharma. All right, when you have a drug that doesn't have a really high therapeutic index, what happens? Well, this is enfortumab vedotin or Padcev. This is a Seagen and possibly soon to be Pfizer drug. That's also an antibody drug conjugate. It delivers a payload called MMAE to cells that express a target called Nectin-4. Well, one of the challenges is that skin expresses Nectin-4, and skin is also very sensitive to MMAE. So this is the drug, this is the study that got this drug approved. You can see it's not overwhelmingly better. The overall progression-free survival is 1.8 months. This is projected to be a $20 billion drug, remarkably. This is what happens to patients' skin. So the median number of cycles that patients can tolerate of this drug is only 4, and then they have to come off the drug. So desquamating rashes and other significant rashes are the #1 cause, but neuropathy and bone marrow toxicity is also quite significant. So clearly an evolution, it's better than the chemotherapy, but lots of room for improvement. All right, challenge number 5, how are we doing with time? Pretty good, is what does best in class mean? Because a lot of drugs, a lot of the things we hear about are, there's a drug out there, it's doing great, it's making lots of money, ours is better, we can supplant it. And sometimes that's absolutely true. So this is Revlimid or lenalidomide, and you can see $12 billion in 2021. That's coming down a little bit, and I think it's either off patent or it's soon going to be off patent. And so the idea is, let's come up with better versions of this drug. This, you heard a little bit earlier about E3 ligases and how they can then lead to degradation of target proteins. The way that these drugs, these IMiDs, work is by binding a complex, including a protein called cereblon, and then leading to the degradation of these target proteins. And it turns out IKZF1 and IKZF3 are transcription factors that are very, very important in multiple myeloma. And that really helps explain the activity. And it turns out these drugs can also degrade other proteins that may or may not have some importance. All right, so that said, the main toxicity of these drugs is on target, off tumor bone marrow toxicity, right? So basically the drug's doing exactly what it's supposed to do. It's lowering IKZF1 levels, but it's not doing it in the tumor, it's doing it in the bone marrow precursors, and that then leads to low white blood counts and low platelet counts. And that limits the amount to which the drug can be escalated in dose. It limits the ability to combine it with other things. And for some patients, they just have to come off because they can't tolerate it. All right. So we got interested in this. My lab had a significant effort in T-cell lymphomas, an area of true orphan. Those are orphans of orphans. Because these diseases are actually very addicted to IKZF1. So you would think lenalidomide would work quite well, and it doesn't. It was tested by Frank Morkenhauser. And it doesn't work. Median progression-free survival, 2.5 months. So what's up with that? Well, we started looking at more potent versions of lenalidomide, like this drug pomalidomide. And this is proteomics, where you just take all the proteins and you just figure out how much does treating with the drug make the protein go away. And this is the relative abundance on a log2 scale, and this is the p-value for the change. And you can see lenalidomide gets IKZF1 down to here, pomalidomide gets it down to here. So pomalidomide hasn't actually been studied in this disease, but it's only slightly more potent. What we found actually is why would pomalidomide work better? Well, it turns out that the amount of cereblon that's within the tumor cell actually determines the extent of degradation of the target. So if we looked at cell lines of T-cell lymphomas that had very low cereblon, they had almost no degradation of IKZF1. But if we looked at cell lines that had very high levels of cereblon, they had complete degradation of IKZF1 and were much more sensitive to these drugs. So you know you can increase the degradation by having a more potent compound. So fortunately, BMS Celgene had been working on much more potent versions of these drugs. And so had a company called C4, which is here in Boston, founded by some friends of mine from Dana-Farber. And so this is what they call a cell mod, CC92480. We got super excited. We partnered with them. These things are active with IC50s in the 10 picomolar range. So for those of you who care about that kind of thing, that's amazing. So we went ahead and tested them. And here at equimolar concentrations, basically the x-axis is if you have a certain number of cells, if you add the drug for each one of these cell lines, how much is it killing? So 10 to the minus 1 would be you killed 90% of the cells, 10 to the minus 2 is you killed 99, 10 to the minus 3, and so on. And as you can see, lenalidomide only works in a few, pomalidomide has better activity, and this drug just slaughters them, right? Many of these are 99% or greater reductions in the number of cells in treatment with that drug. And so that was really, really exciting. If we put this into a xenograft model, so now we've taken a T-cell lymphoma, we've actually xenografted it orthotopically, it's in the spleen, it's in the bone marrow, it's in the lymph nodes. You can see 10 nanomolar of this drug, IKZF1's almost gone. These are the spleens of the treated mice, those are Lilliputian, and in 7 days of treatment, you've got 90% plus reductions in the amount of tumor, and of what's left of the tumor, most of it's actually dying. So this is super exciting. This is going to make a huge difference. No, it's not. And why? Because of therapeutic index. So I just told you that the amount of lenalidomide that you can give to somebody is limited because it does exactly what it's supposed to do, but in the wrong cells. So if you make a more potent molecule, that's also going to do exactly what it's supposed to do, but in the wrong cells. And it turns out, this is the data from C4, they couldn't escalate the drug dose beyond 50 micrograms per day. And so they couldn't see any more effect than you see with lenalidomide or pomalidomide without patients getting very low blood counts. So completely predictable in retrospect. And so I think focusing on therapeutic index is probably the most important lesson I've learned. And then the last one I want to go through is the technology trap. It's just so cool to see new technology and think about how that could transform things. So this is minimal residual disease and the concepts of minimal residual disease. My lab has been very interested, or was, in hematologic malignancies, and there you can get a lot of patients into remission, but you can't cure all of them. So they get down to the state where they've only got a very small number of cells, somewhere below 10 to the 9, but probably many of them below 10 to the 6 cells. So how can you figure out what to do with those cells and how to eradicate them? So we published a review with my colleague Scott Manalis, who's also at the Koch, a few years ago, talking about this is what we do now for minimal residual disease, right? We take patients who have a tumor. This is mostly in the heme setting, but increasingly in the solid tumor setting. We treat them until they're in remission. Until they're in remission, and then we either continue the same treatment indefinitely, or we just stop. Right? So for diffuse large B-cell lymphoma, you get your 6 cycles of R-CHOP, you're in remission, we just stop. And we just wait. Is it going to grow back or not? But what if there was a way in which you could sample very, very small numbers of cells, and then you could actually test those cells, very similar to what we heard in previous presentations, against different drugs, and then you could iterate. All right, so these drugs are sensitive, these cells are sensitive to drug X, treat with X for a while, if those cells go away, but other cells come up, then you treat with these, and so on, and maybe you could eventually get to cure. So how would you do this? Well, you need technology. This is Scott and I presenting this technology at a Koch Institute forum. Scott has been studying for a long time. He's a brilliant physicist and bioengineer, actually how cells grow. So when we talk about cell growth, mostly people interpret that as 1 cell becomes 2 cells, becomes 4 cells, but cells actually have to grow. They have to double their mass, and then they divide. So it turns out that cells have very preserved mechanisms, so going all the way back to prokaryotes, that if you perturb the cell and stress it, it stops growing almost immediately, and actually will start to lose mass fairly quickly. So the idea here is if you had something that was precise enough in its measurement of mass, then maybe you could measure this in a very, very short order. So Scott developed something called a suspended microchannel resonator. And this is basically just a diving board. It has a certain resonant frequency, and depending on the mass of the cell that flows over it, it changes that resonant frequency, and then you can measure, based on that change, what the mass of the cell is. And these things can be engineered to be unbelievably precise. Smaller versions of this can be measured to be precise to the yoctogram. Does anybody, except maybe Mike, know what a yoctogram is? No, you shouldn't, because only physicists talking octograms. It's 10 to the minus 24 grams, right? So it's a billionth of a billionth of a millionth of a gram. It's about the mass of a gold atom, right? So you can't have a postdoc flowing one cell back and forth and back and forth and back and forth. You have to serialize this, and so now you flow a cell in. Each one on the top is a different suspended microchannel resonator. It takes a measurement. Then the cell goes into a delayed channel for 2 minutes. Then it goes over another one. It gets measured again. Then another cell comes in, and then it gets measured again. And you flow all these cells through. You get 10 measurements in 20 minutes. And because it's so incredibly precise, that's enough to tell, is the cell growing? Has it stopped growing? Has it started losing mass? You can also bioinformatically deconvolute the trajectories of each one of the cells. So you know by the time the cell comes out to here, do you want to spit it into a well of a microtiter plate and be able to do single-cell RNA-seq or something like that on that individual cell? So cool. All right. This is what the chip actually looks like. These are printed in France. It's smaller than a penny, as you can see. And we could get this to work with 25 microliters of blood. So 1/200th of a teaspoon. I will say that is less blood than this woman said she could get things to work with. I always get a laugh with that joke. Right? That's amazing. All right, so we went on and did massive studies and published Nature papers and did all the things you're supposed to do as an academic, and showed that we could collect cells from mice that were in very, very deep remissions, and then we could test them for different drugs. Working with Alex Shalik at MIT, we could study the states of those cells, and we could then use that to predict what drugs to actually give to eradicate MRD, and we could do that too. So we could actually cure mice that weren't cured if we added on a drug, and I should mention losmapimod, duvelisib, ibrutinib, fostamatinib, all FDA-approved drugs. So all the combinations were with FDA-approved drugs. Again, one model. So it can be done as a proof of concept. We started a company called Travera that's over in Cambridge to do exactly this, and it's unbelievably hard. It's just unbelievably hard. It's unbelievably hard to get the samples, to understand the heterogeneity of treatments. Most patients are getting combinations, so how do you mimic that combination in vivo? How do you get around things that are using the immune system in vivo? How do you understand the competition going forward? If you start a trial, prospective trial of standard of care, if it's no longer standard of care 2 or 3 years from now, how do you get reimbursement for novel therapies, even if you do make a prediction that a patient should receive a particular treatment? And then there's just a field-wide cynicism. And I know Keith had spoke to this point as well. If we're going to move the needle, maybe it's with this. Maybe it's with functional precision medicine approaches on patient samples in rare tumors, but man, it is hard. All right, so to finish, how do we maximize the value of our investments? This is how we think about drug development. It's pretty obvious. You start with a whole bunch of information. You decide you want to go after a target. You start coming up with composition of matter. You actually make a drug, and you bring it to clinic, and you have off-ramps at every single point, right? And you try your hardest to avoid issues around sunken costs, which is really, really hard. But I also want to highlight this data. So this is data that came out in the New England Journal of Medicine last week. Keytruda is as I said, remarkable drug indicated for many, many different diseases. But it takes a really, really long time to do all the studies to get to the point where you have shown across all the different cancers that you've maximized the benefit. And I'm not even talking about adjuvant therapy, or if we ever got to preventative therapy or anything like that. It takes decades. So the NCI scorecard came out recently, and it talked about how over the course of the pandemic, only cancer actually had lower mortality. Every other disease type, cardiovascular, infectious disease, everything else went up, but cancer actually came down. And the major advances in cancer were in lung cancer, right, due to both PD-1 blockade and tobacco taxes, and in cervical cancer, I would argue due to our HPV vaccine and comparable vaccines. And in women under 30, cervical cancer is down like 80%. It's unbelievable. Endometrial cancer was called out as a cancer where there has been no advances. There have been no advances. This study then came out last week. This is exactly the same design as the one I showed you in lung cancer that was performed 7 years earlier. And it's chemotherapy standard of care plus placebo versus chemotherapy plus Keytruda. And look at those curves. You could drive a bus between those curves. This is in the subset of patients who have mismatch repair defect. It didn't do as well in the patients who didn't have mismatch repair defect, but it was still very, very significantly different. So writing textbooks 10 years into a therapy. So as we think about where we're going with the next new shiny object, there are still therapies out there we just don't know how to get these funded, especially closer to loss of exclusivity for certain drugs. It just becomes extremely challenging to get those studies done. So I will summarize by saying most ideas, including mine, have fatal flaws. Ferreting out the good ones is really hard. But figuring out the best use for current and hopefully massive future investments is extraordinarily important, and it's going to take a room full of people like this one to get there. So thank you very much.
We can take just a couple of questions, if anybody has any. If not, we'll actually give you a break and let us pan over the room. But, oh, yep, no, we have one over there. Hang on.
Thanks for the wonderful talk. I'm regular. I used to be the 65 Business Development Program at Williams a long time back. My question is, if you look at the vast amount of drugs that have not worked, it seems like if you include some of the new technologies that are there, there's more gold to be gotten by mining those drugs that have not worked and by making the therapeutic index better, for example, versus the new drugs. However, the incentives don't seem to be there for any pharmaceutical company to do that in a way that will reward them.
Yeah, I think that's a very reasonable hypothesis. And it's impossible for me to tell where resources would be better put going back through the millions of molecules that have been generated at Merck. You know, we literally have old books, you know, the chemists wrote shorthand and trying to figure that out with some kind of AIML and then putting all that together and then redoing screens and so on versus new target discovery. I agree with you. When you've got hills that big, there's gold in them. But it's a super challenge. There are ways to take a molecule that has activity, but maybe a problem with therapeutic index, and then think about how you might iterate on it and generate new IP, and then financially might be interesting to a pharmaceutical company. And there are good examples of that type of thing in the past. I mean, lenalidomide is that, right? Thalidomide was active in multiple myeloma, and the IMiDs were created on that basis. So I think it depends, you know, which one of those approaches is more exciting to you.
David, great talk. My name is Todd Astor. I'm a transplant pulmonologist. And so I think there are some really strong parallels between oncologists and transplant physicians with how they look at therapeutic index. So I'm wondering the perspective of therapeutic index as it relates to regulatory approval compared to reimbursement and then adoption, and how you look at those perspectives differently from a drug development standpoint.
Yeah, it's a great question. That's something I'm still learning, but I am learning that I used to think you got the drug approved and you could, you know, have the celebration party, and that is just not the case at all. It's not the case in particular with HTAs, and you know, when Keynote 189 comes out or the endometrial cancer comes out, there's an army of people who are going to, each of the European countries have their own approval, right? So in Germany and in Switzerland, and you have to present data that shows not only the overall survival benefit, but also that this is going to be something that, you know, people should be interested in paying for. And if it's really toxic, but it creates an overall survival benefit, it's not going to get the same reimbursement as something that's less toxic. I don't know if that answers your question, but we are certainly seeing it.
So do you figure that into your design drug design? That's the very beginning, what you decide to bring to later stages of development?
I mean, I wish I could predict so well, right? 95% of my ideas are going to fail. So if I had the discriminatory ability to understand how much tox that drug was going to cause versus another one, that would be fantastic. Certainly, if there's something that we see as a potential fatal flaw of on-target tox, we'll probably deprioritize it.
Over here. Hi, Chris Hughes. So is drug synergy not real, or have we just not trialed drugs that act synergistically?
I cannot answer that definitively, although my wife has told me that everything I say sounds like I'm saying it definitively.
Not very compelling.
I will say that the suppositions that we make about human trials are irrational based on the synergy that we see in preclinical settings. So in two-plate dish or organoids or something, you're exposing the tumor cells to a constant concentration of both drugs. That obviously does not happen, right, in human beings. That's just one example where why would we think we would see that synergistic effect? The other is much of the synergy that we define in preclinical is by taking each of the drugs at multiple different doses. And where we see a sweet spot of synergy is actually if you lower the dose of each drug below its most effective dose, right? And then you see this synergy. Well, most of the time, that synergistic overall effect is still less than if you used the maximum tolerated doses of the two drugs together. You'd get better efficacy, but you'd also get more toxicity. And we end up in the patients giving that higher dose. So I think those two fundamental principles are the ones that I lean on most when I make the statement that we just don't believe in human synergy until someone proves it.
Right. Well, I guess the flip side of that is if you could use a lower dose of each drug and get the same efficacy, you might actually reduce the toxicity and open up your therapeutic window a little.
If it was a lower dose that also maintained an adequate level in the tumor cells over the time period, yes, so absolutely.
Well, it's really a reaction to what you're saying is that the toxicity is largely, for the most part, for most of these drugs, a factor of the scheduling. And you can overcome a lot of the toxicity with clever scheduling. I don't see very much clever scheduling out there in drug development coming from big pharma. Why is that?
I think to some extent we're not thinking hard enough. So ADCs, tremendously active drugs, but they completely ignore one of the fundamental principles of dose density that Larry has brought forward for over the course of decades, which is hit the tumor and the normal cells hard, and you get a therapeutic index because the normal cells are better at repairing once the drug is gone. An antibody-drug conjugate circulates in the blood the whole time, and you're constantly bathing the normal cells in the conjugate. There are moves, for example, by a company called Bicycle that's developed a Nectin-4 peptide, much, much shorter half-life, and they see less on-target toxicity. It's still a small number of patients, but I think that's an example where somebody's actually thinking about it.
I actually have one final question on behalf of the entire room. Put your Merck hat on for a moment. How do we work with you? How can we connect with you? How can we collaborate with you? How do we get some money?
Yeah, so I think, so now that I have the Merck hat on, I'll say things like, the people at Merck are absolutely dedicated to improving the outcomes of people with cancer. How do we work with you? I don't know, email me. Throw me an opportunity that's mutually beneficial. And I can say mutually beneficial, not solely like that's going to make us money, that's going to, there are many, many other things that are good for Merck. We have, for 40 years, given away free ivermectin for river blindness in Africa. This is something developed by Roy Vagelos, billions of pills for free. And we tout it, and we're really proud of it. And we can recruit people because we do things like this. So that's good. I would argue part of the Sanofi thing was they didn't want shame out in the public, right? So shame is a powerful force for getting companies to do things, not with Merck, but with other companies. So, but I think, let's talk as colleagues about opportunities. I think that huge database that we have, I'm going to talk to Keith after the meeting of Keytruda outcomes. As we get closer and closer to loss of exclusivity, the opportunity to leverage that for really meaningful advances across the entire world becomes greater.
Thank you, David. Thank you so much.